Okay, hopefully we're recording now. I haven't tested out sound yet, so no doubt it won't work. Now that'll confuse people even more now that turn up. Surely they're not going to. Alright, let's get started. So today is a very interesting, I find it an extremely interesting lecture, and it actually puts a lot of things together that you've been working on. So suddenly things like PCR make a bit more sense in applied terms. So as you know from your outline, we're going to be talking about recombinant DNA techniques, and if we really want to know what we're talking about there, it's just as the name suggests, we're joining DNA, so recombining them from two different sources, okay, to create something, usually something new. So the little diagram I've got up on here is a classic example of recombinant DNA usage to create these aquaria fish, these glue fish, which are actually commercially sold in some countries, created by some researchers from the National University of Singapore, and they're one of the first transgenic animals. Why are they transgenic? Because the DNA that's been recombined, that's been joined together, is obviously some DNA from the zebrafish, but also some DNA from other fluorescing animals. So where's my pointer? The green one here would have had probably the green fluorescent protein from a jellyfish, isolated from it and joined up and with the DNA of the actual zebrafish, and these other fluorescent fish that we can see would have had some genes that code for different types of fluorescing proteins placed in them and put in. So that's obviously showing it in a very novel kind of use. They're an aquaria fish, but the whole point of these green fluorescing proteins usually stems from their uses as markers for other kind of scientific questions, and we'll touch on it a bit later. But as long as you know that recombinant DNA is just the joining of those two different sources. So the ability to manipulate DNA like this is it's an amazing kind of breakthrough. I mean, it's been around since the 1970s, when I think a couple of researchers, maybe at MIT, or one of the US universities at least, patented the term of recombinant DNA technology. But it's just made a huge explosion. So when I say it's led to a new genetics, we're no longer really worried or concerned too much with having to selectively breed or induce mutations through experimentation, or even follow phenotypic traits. We can actually directly go in and alter things in, as I said, a far more direct way, and that allows such a flexibility that it's led to this kind of biotechnology scientific revolution. So here once again, on just the image there, you can see that this is a tobacco plant, which is one of the plants that's traditionally been used as a tool or an experimental model for a lot of recombinant genetics, and it's got, once again, the green fluorescent protein from that jellyfish spliced in, so it's fluorescing. So if we start off with some of their particular uses to put it into context, and these are countless, and I'll show a couple of videos in a minute which are really quite interesting and kind of hopefully showed some of the diversity. But just so there's some in a slide format here, we can see from the food industry right through the pharmaceutical industry, medical research and agriculture, aquaculture. They've been used for many different applications. In 1990, recombinant chymosin was used. So those of you who cook or know enough about cooking or even like cheese, I don't like cheese, but rennin is basically made from chymosin, which is an enzyme that used to be extracted from the stomachs of cows and used to set cheese. Alright, lovely, just makes me love cheese even more. But anyway, in 1990 we could then recombinantly produce that same enzyme, that same protein, and no longer have to actually isolate it from a cow's stomach. And reasons why? Well, it's usually cheaper and much more efficient. Okay. Golden rice. These are some of the food industry's attempts to solve the world's problems. Golden rice, it promotes beta-carotene biosynthesis and reduces vitamin A deficiency. So it's actually designed to try and be fed to populations in the world where vitamin A deficiencies actually lead to multiple deaths in young children, etc. And there are bananas, just the same, that have been genetically modified, which is really what we're talking about here, through recombinant DNA techniques, so that they can actually produce more of vitamin A. The pharmaceutical industry, one of the classic ones is the production of insulin. So whereas it used to be kind of taken, isolated from pigs, etc., now it can be recombinantly produced in bacteria. So we get the bacteria to produce the human insulin, isolate it from the bacteria, and we have it. And we no longer have to worry about killing or taking it from other animals. Same goes for growth hormone to treat, well, insulin was obviously to treat diabetics, type 1 diabetics, but recombinant growth hormone, people who have growth hormone deficiencies may require that, and it's much easier to produce these things recombinantly. The list goes on and on and on and on, and in medical research we could have a whole lecture just on the applied uses. Some of the interesting ones for agriculture or aquaculture. Well, agriculture, you hear all the time the use of genes for making things resistant to common glyphosates, so Roundup, etc. That gives it a bit of a bad name, the old recombinant DNA technology. However, there are in things like corn, BT corn, which has been around for a while in the States, where they've spliced in poisons from insects and into the corn, so the corn produces them itself, and then some of the classic pests of corn are actually killed when they try and eat the corn. Okay, so there are definite ways of kind of getting around some of the negative impacts, etc. In terms of aquaculture, because this is an aquatic molecular course, Aqua Bounty is an interesting one. It's a company that formed, I think, out of Canada. But what they've done is taken a Chinook salmon growth hormone gene, and they've actually spliced it in and recombinantly joined it up and put it into Atlantic salmon, and it allows the Atlantic salmon to grow a lot faster. And you can't see too well here, but this red line here is the Aqua Advantage salmon, so it's got the Chinook growth hormone in it, and we can see, well, at 700 days from first feeding, we're looking at a salmon that weighs close to 6 kilograms versus the standard Atlantic salmon, which after 900 days is only approaching 4 kilograms. So that's a significant advantage. Okay, and you can see they've tried to do a comparison here between the smaller standard Atlantic salmon of the same age to this, what do they call, Aqua Advantage salmon. Okay, so the growth hormone is from the Chinook salmon is obviously very efficient when placed in this Atlantic salmon. Okay, why would you want to do that? Well, all sorts of reasons. Commercially, you're going to get your fish to market a lot faster, but that's obviously looking at it from the aquaculturist's point of view. But if you take it from an environmental aspect as well, you're likely going to have to feed less to this animal, because you can, whilst you might be feeding the same amount, it's for a much shorter period of time. So therefore you're minimizing your resources that are put into that. And of course, we probably know that for Atlantic salmon, there's a lot of feed that comes from wild fish. So it's a resource that's constantly in the public eye for being possibly depleted. Okay, so fish oil, fish meal, etc. So you're minimizing those effects. Not to mention they talk about health aspects as well. But if you are interested, just typing in something like Aqua Bounty will give you plenty of, most likely, propaganda, but some also useful information around what they've done to produce that.
Are they doing this here?
No, so Aqua Bounty is, as I said, a Canadian company set out of, it's probably registered in the US, but the problem is, as you know about any transgenic animal, there are huge public concerns. So these salmon can't obviously be grown in a sea cage. They have to be kept in a very isolated tank environment, and from memory, they were actually growing these in Panama. So somewhere that Atlantic salmon aren't really going to be a problem if they escape from their tank environment. But I mean, even then, they're made so that they're all female stock anyway, so they're very unlikely to be able to breed.
Wasn't it just last year the government, Tasmanian government, put it on like a complete ban on the GM?
Well, that's right. So like I'm speaking in a global kind of sense here. Tasmania, it's a bit of a problem. Yeah, it's not just last year, it was quite a bit before that they've got their moratorium or whatever that they talk about with complete ban. It's very probable. Yeah, I won't weigh in on the argument, but there are definitely pros and cons, some great benefits, and those benefits may far outweigh the cons. Who knows? All right, so just some other little techniques here. We'll see if we can get these videos, because these are quite interesting to watch. Hopefully we won't have too many issues here. Hopefully we have sound. Probably end up with some
Curious Droid presents
full screen.
7 freakish genetically modified animals. Over the past 30 years,
Anybody
biotechnological
here?
developments have allowed scientists to alter the genetic makeup of bacteria, plants and animals. Here are 7 of these such experiments, but they represent just the tip of what will become a
Let's
massive
just pause
iceberg.
back
At number,
here. So
but
what
they
we're looking at
represent-
what we're looking at here is some mice, transgenic mice, and they've actually got that green fluorescent protein in their genome as well. Okay, so under UV lights you can see they're glowing. Why would you want to do this? Well, usually you put that green fluorescent protein in along with some other things that you may have altered, so that instead of having to go in and do a PCR or some other technique to detect whether you've managed to incorporate a change, you may be able to just look and see that it's fluorescing. So if you know it's fluorescing, you know that you've introduced the other change. So it's used as a marker. Okay.
bacteria,
Then
plants
move
and
on.
animals.
This is a pig that's been made to fluoresce as well. We're not under UV lights here, but if we did, you'd see that this one would be glowing a strange color as well. Okay.
Here are 7 of these such experiments, but they represent just
All right,
the tip.
this is a double-muscled cow. This is actually a natural occurrence in Belgian blues. So there's a gene that regulates muscle growth, myostatin, and if it has a mutation, and this particular breed has a natural mutation that makes it non-functional, then the muscle growth isn't controlled, and you get these crazily muscled cows. And if you search the internet enough, you'll find that you'll see that there are even young kids that are naturally born with this this mutation in the myostatin gene, and they'll be ripped. Okay, so this is one way, and we're not interested so much in making crazily animals of the muscle, but the benefits there come to things like trying to treat diseases, genetic diseases like muscular dystrophy, or even just old age where you lose muscle tone. If you can go in and you can alter the myostatin gene with the use of recombinant technology, then you possibly can increase muscle mass and get around some of those problems.
Of what will become a massive iceberg. At number 7, we have the sudden death mosquitoes.
Can you hear it over
Dengue
there?
fever is a mosquito-borne virus that causes 2.3 million infections and over 25,000 deaths per year worldwide. As yet, there is no cure. But to deal with this, scientists have created what are called sudden death genetically modified mosquitoes, which are released into the wild and breed with the indigenous mosquito population. These then pass the special gene to their offspring. This special gene means that the baby mosquitoes die before they themselves could breed, thus reducing the number of mosquitoes that could go on to infect people. Trials in specific areas have showed up to an 80% decrease in the mosquito population for up to 3 months after the initial release. Number 6. Glow-in-the-dark cats. The glow-in-the-dark cat was developed as a way to fight the feline immunodeficiency virus, or FIV, which is related to the human version of HIV and typically affects feral cats. In 2011, American and Japanese scientists inserted genes into cats to help them resist FIV. In order to mark the cells more easily, they also inserted a green fluorescent protein into both genes transferred to the feline eggs. This protein, which is naturally produced in jellyfish, is commonly used in this area of research to monitor the activity of the altered genes. This let the scientists examine how the resistant gene developed within the cats by examination under a microscope. The modified cats are always normal during the daytime, but sometimes they glow at night. Number 5. The web-spinning spider goats. When it comes to natural goods, flexible and strong spider silk is incredibly valuable. Spider silk is, pound for pound, 5 times stronger than steel and far more elastic than rubber. Normally, getting enough spider silk for these applications requires a large number of spiders. However, spiders tend to be territorial, so when the researchers set up spider farms, the spiders killed each other. So they inserted the silk-making genes into the mammary gland cells of goats. The cells then make the extra protein to make the spider's silk. The milk can then be collected and the protein is extracted and spun to make the spider silk thread. Number 4. Singing mice. Sometimes there is no defined end result for the genetic tweaking involved, and this is what happens with the Japanese Evolved Mouse project. It's a bit like adding some extra genes and let's see what happens when the results of the experiments are allowed to breed. One morning, whilst checking on a new litter of mice, researchers discovered that one of the baby mice was singing like a bird. They focused on that mouse and have now bred over 100 mice that can sing, but it doesn't end there. They noticed that when normal unmodified mice grew up around the singing mice, they too began to use different sounds and tones, almost like a simple language. The project's goal is to artificially speed up evolution, but we may have just created a super talking mouse that will one day go on to take over the world and experiment on humans in the future. Number 3. Popeye pigs. For all you meat eaters out there that love bacon and hate vegetables, Japanese scientists have taken pigs and added the gene from spinach. This creates a healthier meat with a lower saturated fat content and contains proteins that are found in the spinach leaves. Dubbed the Popeye pigs because of the spinach, these pigs have been inserted with a special spinach gene that converts the saturated fat into unsaturated fat. Although the pigs have been cleared of any health complications, their announcement was met with a public outcry. Maybe a better solution is just to eat more greens with your organic pork instead of trying to create pigs that do both. Number 2. Cows that produce human milk. Babies could be fed on human milk produced by cows in the wake of the latest genetic modifications. Scientists have created cattle that have been given human genes to make their milk contain the same nutrients and fat content as breast milk. However, human milk differs from cow's milk in several important ways. It contains high quantities of nutrients beneficial to the baby's growth and also special immune system antibodies that are specific to that mother and baby, which could never be replicated using this method. Number 1. Glowing zebrafish. Glowfish is a patented brand of genetically modified fluorescent zebrafish with bright red, green and orange fluorescent colours. Though it was originally created to show the presence of toxins in water, it was the first genetically modified animal that has gone on to be widely available to the public as a pet. In 1999, scientists at the National University of Singapore were working with a gene called the green fluorescent protein, or GFP, originally extracted from a jellyfish that naturally produced a bright green bioluminescence. They inserted the genome into the zebrafish embryo, allowing it to integrate with the zebrafish's genome. This caused the fish to be brightly fluorescent under both natural white light and also ultraviolet light. The idea was to develop a fish that would detect pollution by glowing in the presence of environmental toxins. Thanks for watching, and I hope.
Okay, so you can see that there's some pretty interesting little tools there. And I say tools because they're not all just novelties. The glowfish actually was designed for that purpose of being a biomarker for heavy metal poisoning and other pollutants in the water. Okay. Let's get back. Now this other one, and we'll quickly have a look at that, and then we'll show you how this all can occur.
Welcome to Top 10 Archive. Think it's scary that geneticists are altering the genetic code of animals to make them glow in the dark? Then you'll love knowing that you've probably been ingesting and living with genetically modified plants without even knowing it. While some have practical purposes, others aren't quite what we consider a necessity. Number 10. Bug-killing corn. The corn borer is a nightmare insect, specifically for farmers specializing in growing maize. Thanks to genetic modifications to regular corn that may be in your refrigerator right now, the corn borer is no longer a big issue. This genetically altered crop produces the Bt protein, which acts as an insecticide against the crop's biggest enemy. To keep the borer from evolving a natural immunity against the protein, crops are required to be a ratio of 80% of the GM plants to 20% of regular corn. Introduced in 1996, the modified corn has benefited farmers in Illinois, Iowa, Minnesota, Nebraska, and Wisconsin, saving an estimated $7 billion in total. Number 9. Non-browning apples. Ever cut into an apple and set it aside for a minute? By the time you turn back to your fresh fruit, it's already starting to look a little worse for wear. The browning process, or oxidation of the apple, is very rapid. But there's a cure-all in a genetically modified version produced by Okanagan Specialty Fruits in Canada. These specially treated Arctic Golden and Arctic Granny apples are developed through a gene splicing process that negates the chemical reaction responsible for the browning process. As of 2015, the U.S. Department of Agriculture decided to deregulate the engineered apples, starting a long road to consumer consumption in the future. Number 8. Flavor Saver Tomato. It's hard to ignore the very infomercial-esque name of this genetically modified tomato, and there's a chance it had something to do with the eventual failure of this long-lasting version of the popular vegetable, or fruit. Flavor Saver tomatoes, developed by now-defunct Calgene, were created to have a longer shelf life and stronger exterior, which would allow them to be harvested without worry of damage to the product. In the end, though the tomato proved to last longer, it still retained its softness and had to be handled as any other red tomato. An advancement in flavor down the line caused a jump in Flavor Saver pricing, and over time, both Calgene and its staple product vanished. Number 7. Super Bananas. Despite popular belief, household bananas are not genetically modified, despite looking nothing like their wild counterparts. That doesn't mean there aren't GMO versions of the fruit out there, though. In fact, scientists have moved forward in creating what is being dubbed a super banana. Though they look like their untouched common household fruit, the engineered end product has increased levels of vitamin A. The modification was done in hopes of helping the thousands that succumb annually to fatal vitamin A deficiencies. By 2020, researchers hope to have the product in Rwanda, Kenya, Tanzania, and Uganda, pending trials in the United States. Number 6. Cancer-fighting tomatoes. Though the tomato has been associated with helping stave off prostate cancer, a series of genetic modifications have produced something that may be quite a bit more beneficial: the purple tomato. Initially grown in a Leamington, Ontario greenhouse, has been altered to produce a higher level of anthocyanins, an antioxidant typically found in blueberries, plums, and blackberries. Anthocyanins is not only responsible for the purple color of these fruits, but is also believed to fight off cancer. Initial testing of the purple tomato performed on mice had shown a lifespan increase of 30% than those on a diet of plain red tomatoes. Number 5. Pollution-fighting plants. High levels of pollutants are a growing concern for much of the world, and part of the answer may be in an unexpected source. Scientists and researchers have begun to look at grass and trees as an alternate and inexpensive means of battling toxins. But to do so, a little genetic modification will be needed. The process of using plants to cleanse the environment is not a new one, but phytoremediation is considered too slow and cleanup is too costly. The engineered plants have shown quicker processing of the carcinogens, leading to the hope that phytoremediation will be a suitable means of pollutant cleanup in the future. Number 4. Venomous cabbage. It's going to be difficult to market cabbage spliced with a scorpion venom-created virus, but that's one of the many genetically engineered plants on the horizon. While we're conditioned to fear getting stung by a scorpion, the venom being interlaced with heads of cabbage will not be harmful to humans, or so we hope. Instead, the target in this scenario is caterpillars. The scorpion makeup will rapidly introduce a nerve poison into the dining caterpillar, causing it to become paralyzed, allowing the virus to kill it. Early trials showed that the engineered virus was fatal to dozens of species of butterflies and moths, leaving many to question just how ecologically safe venomous cabbage really is. Number 3. Genetically modified trees. Trees are far from exempt from human tampering. Take, for instance, rubber cork trees and trees used for paper. For the rubber cork tree, it was all a matter of consumer desire. Wine enthusiasts, displeased with the introduction of the rubber cork, sought an alternative. Sabic Innovative Plastics modified the rubber tree, which rubber corks were made from, and spliced them with the cork tree. The product was a tree that produces a cork that falls in between rubber and cork. In regards to paper production, scientists at University of British Columbia, University of Wisconsin-Madison, and Michigan State University have engineered trees that are easier to break down for the creation of paper and biofuels, leading to less pollutants during production. Number 2. Super carbon-capturing plants. As carbon-producing life forms, we humans need a counterpart to help absorb the 9 gigatons of carbon that we produce annually. Of that 9, approximately 5 is absorbed by plant life, leaving 4 gigatons of carbon left to pollute our atmosphere, leading to fears of global warming and the greenhouse effect. To take care of that remaining 4 gigatons, researchers have turned to genetically engineering plants, focusing specifically on creating bioenergy crops with large roots to capture and store carbon. Compared to the leaves, branches, and flowers of plants, the root can house carbon for a much longer period of time, greatly reducing the amount left over. Number 1. Landmines detecting plants. While wars may end, weaponry used during them don't just vanish. Specifically speaking, landmines are still found during times of peace from wars that have long ended. Danish researchers at Aresa Biodetection claimed in 2004 to have developed a plant that changes color when introduced to compounds often leaked from landmines. The current cost of detecting and clearing landmines is upward of $2,000 per mine, but the introduction of such a plant could reduce the cost greatly. Coupled with new drones that are designed to seek out signs of landmines, these plants could help save both lives and money. I know, but even if they could come up with a bug-killing, non-browning, flavor-saver, cancer-fighting, pollution-fighting, carbon-capturing, landmine-detecting super tomato that tastes like a banana, just saying, you still can't get a 2-year-old to eat it. Call it the Super Tomana.
All right, so you can see that there are some very important uses and some that are quite trivial. Okay, so but nonetheless, it's just a bioengineer's dream to be able to use recombinant technology. Okay, so how do we go about doing this? How do we go about combining genes from different sources into one? Well, the traditional way and the way that we've been using up until quite recently, and probably still are for most of it, is the use of restriction endonucleases. Okay, so these are like molecular shears or scissors. So they're endonucleases. Who can tell me the difference between an endonuclease and an exonuclease? Yeah?
Endo's inside.
Yeah, that's right. So the endonucleases actually just search out a specific sequence within, say, a lineage or a strand of double-stranded piece of DNA, and will cut it there. Exonucleases, which you're probably familiar with from the hydrolysis probes from the qPCR assay, they digest from the ends. Okay? So that DNA polymerase for the Catman or hydrolysis probe assay that has exonuclease activity, it uses that to displace the probe from the DNA sequence. Endonucleases obviously seek out a specific sequence within. So they're isolated from bacteria, and the bacteria naturally have these to fight off and destroy viral DNA that may be trying to get incorporated into the bacterial genome. Why don't those nucleases attack the actual host? Well, they can recognize whether or not there's methyl groups attached to the DNA. So the host has methylated DNA, and it basically protects it from these endonucleases. So basically, you just have to think of them as molecular scissors. So they cleave at specific sites. You can see here. Here we have a recognition sequence, and this particular endonuclease, which is named EcoR1, comes along, recognizes that, and it can digest it and cut it like scissors. Where it has these overhangs, we call them sticky ends, okay? Cohesive ends. And that's important for how we go about recombining, so how we go about rejoining the cut DNA a bit later. But not all restriction enzymes, as I'll commonly refer to the endonucleases, cut and leave sticky ends. Some of them cut and leave blunt ends. And you can see already that combining blunt ends is a lot harder than combining the sticky ends, because the sticky ends have that actual base pairing to aid, whereas the blunt ends where there's no real base pairing that's going on there, you're just joining two ends with phosphate and hydroxyl ends, 5 and 3' sitting over the end. Okay. I said that one was for EcoR1. What does EcoR1 mean? Well, they have this common terminology where E is usually stands for, the E in this case stands for the genus, so Escherichia, from Escherichia coli, which is the actual bacterium that this endonuclease was taken from. You've got R, which is just a particular strain, and the one first identified order in that bacteria. You'll notice that there's lots and lots of different endonucleases, and most of these probably were identified in the 70s, I suppose, where biochemistry was at its absolute prime, so before the molecular side of things had taken off. But scientists had already isolated, or biochemists had already found many of these enzymes that can actually go through and recognize sequences and cut them, as we can see here. Okay, now if you think about it, how often is it going to cut? Well, it depends upon, at least for the ones that have a sticky end sequence, it all depends upon how many bases are formed in that recognition sequence. So if you've got 4 4 bases pretty much. If you have a 6 base pair recognition sequence, you're going to probably get on average 4 to the power of 6 nucleotides along before that sequence will appear within a random genome cut. So the point to get across there is the longer that recognition sequence, the less likely you are to, by chance, meet that or encounter that particular sequence in the genome. So if we just take a cut piece of DNA here that's been cut with our restriction enzyme, so our endonuclease, we can see that we're left with our phosphate and our hydroxyl group on either strand, and if it was allowed to sit there and come into contact with the DNA ligase, well then it would just recombine and rejoin up. This is a circular piece of DNA, it could be linear. However, to stop them just recombining when we throw in ligase, we usually go in and we treat with a phosphatase the ends of the cut DNA. So basically give them a couple of hydroxyl ends. So we're digesting off the phosphate with phosphatase. And then this won't spontaneously recombine even with ligase to reconnect. Okay? Because once we cut something, we really want to keep it apart as much as we can. Now what we can then do is we can prepare our DNA insert, and we can have the ends, so we have our hydroxyl and our phosphate group, and then when they're put together with the ligase, we end up with them kind of joining with a couple of nicks on either end where they don't quite match up. Because obviously the phosphate will combine with the hydroxyl group of one and the other, but the two hydroxyl groups aren't going to come together. And then we use another enzyme which comes along and just quickly repairs that little nick. So that's just to get across the fact that how we can kind of control what we want to be pasted back in. Because if we made a big mix of this and we hit our insert, we're going to get a lot just joining back together before it's had chance for our insert to go in. So we're trying to maximize the pasting ability of our insert there. And DNA ligase is the one to really remember for that joining. Okay, so once we have actually made our recombinant DNA, how do you go about getting lots of it? We know now quite a bit about PCR, so we know how we can use PCR and DNA polymerase to not randomly, but to exponentially amplify our amplify of interest. But that's not usually what we would be doing in a recombinant sense, that we would be combining with gene cloning. So we're going to be introducing gene cloning now, I suppose. So the PCR we can consider in vitro replication, but then we have our DNA or gene cloning, which is then taking advantage of the replication machinery of another organism. And this we'll probably be talking mostly about bacteria. We're going for time. Okay. So here we have just another representation of how we cut and insert. But what we're looking at here is a plasmid. And you already met with plasmids with Chris Bolsh. I think you have, from memory. So these are the self-replicating circular DNA, usually within the bacteria. So it's a bit distinct from the circular genome. So these lend themselves perfectly as vectors for actually inserting and copying DNA, recombinant DNA. So this particular plasmid we're looking at here, obviously it's circular. We'll explain what some of these kind of terms mean that are representing particular areas of this circular DNA. But at some point we're going to cut it with a restriction enzyme, just as we saw in the previous slide, and we'll then go and insert our piece of DNA that we want to put in there. Okay, and you can see this one's got some sticky ends. It'll go in, and eventually when it goes in there, we'll have a plasmid that has a gene that we actually want in there. And it'll become hopefully pretty clear to you how we can actually put a lot of those green fluorescent proteins and get animals to start glowing in a little while. So if we kind of knock this down to its basic events for gene cloning, we have to obviously have our DNA of interest. We may have had to isolate that from a source somewhere. Say we want to start cloning insulin, well then we really need to go to the original source, extract that DNA of interest. And once we've got that, then we can incorporate that gene into a vector. The one we've just spoken about is that circular piece of DNA forms the plasmid, but there are some others we'll meet in a minute. And then that vector is usually transformed into something that's going to grow, another organism that's going to grow. So commonly be using bacteria, talking about bacteria, but it might be going into yeast, or it might actually be going into a eukaryotic cell. Once that vector's gone in, then as the organism replicates, or the eukaryotic cells replicate, so does the vector, and hopefully you get replication of your gene of interest. So it's easier to visualize in the form of a bacteria or bacterium, because we know that bacteria grow quite quickly. So E. coli, it's what, doubling every couple of hours or something. Yeah. So every doubling, you're getting more and more and more and more and more, and then you can isolate your vector, which in this case would be a plasmid from that bacteria. And you have suddenly without PCR, so without in vitro replication, we've managed to obtain more of DNA of interest. So is that reasonably clear? Just everyone the fundamentals there? Yes? Okay. So here is what we classically see for a very simple plasmid vector. And we probably should become familiar with some of these terms. So this little site over here is a very important one. This is the multiple cloning site. So this is where we'd be trying to put our insert in. So our DNA that we cut out of some DNA that we may have isolated from the source and we want to actually copy. So we cut it out and we place it in here. Don't worry too much about the actual base pairing around here, but you can see there's lots and lots of these multiple cloning sites. Are called multiple cloning sites because it means that you're able to cut with any one of these restriction enzymes from EcoR1 all right the way through all these strangely termed endonucleases, and if you cut with those, you'll be able to insert and combine into that particular vector. Okay. Now, then the other thing is, okay, where has that been inserted? Well, this is actually a gene here that's coloured in red. It's the LAC-Z gene of the lac operon. So you all know what an operon is? Well, I'm sure you do. Well, no doubt we'll refresh your memory in a minute. But fundamentally, that operon is going to code for the production of some genes that are very important to the use of lactose as an energy source. Okay. And by disrupting that gene, by placing in our cut piece of DNA into the multiple cloning site, we're disrupting that gene so it can no longer do its task. What else have we got in there? We've got our origin of replication. So that's where our replication is going to occur. And so you probably, I know that you're familiar with that one because I sat there for part of the lecture and watched Chris explain to you how DNA replicates. So that's where the strands become apart, be unwound, and you would actually have your enzymes coming along and basically copying the DNA, okay, to make so that it can actually replicate. So that replication is important. The origin of replication needs to be there, otherwise your vector isn't going to actually replicate. Then we've got here an ampicillin gene. So this is an antibiotic gene, and another one here, kanamycin. And hopefully it'll become clear why we have those in a minute. Okay. So here are the three features that all cloning vectors should have. So sequences that permit the propagation. So they need to be able to replicate, otherwise there's no real point in having them. They need to have the cloning site. So you've got to have somewhere that you can actually, that's made so you can insert your particular piece of foreign DNA. And usually you're going to need a method for selecting bacteria that house or that have been transformed with your vector. So you need to be able to select those which have your vector from those which don't. And that's where that black operon in its simplest form will come. Or we'll be explaining how that actually is used as a marker. Okay. So here is just showing you what the circular plasmids look like. Okay. So this is electron microscopy. You can actually see circular DNA and plasmids don't just sit there as a nice round circle. They do fold and twist, etc. Plasmids are just one of a lot of different cloning vectors or vectors that we use for cloning. They're the simplest one to explain all of their terminology, theory behind. The other vectors which we'll introduce in a little minute, it's all the same process. Fundamentally, you're using it as a vector, so you're carrying in your DNA and you're having it propagate once it's been transformed to some sort of host. Now, specifically talking about plasmids, they are a bit limited in size of things that you can put into them. So the size of the DNA that you can insert. And that is probably the reason why you might be asking, well, plasmids are so easy to understand and easy to manipulate, why not just use them for everything? Well, they have limitations, and one of those is the size of things that can be put in. The other is the fact if we're trying to express a protein that a gene that codes for a protein that we put in there, it's going into a prokaryote. Usually the plasmid will be transformed into a bacterium, and we know that a lot of the bacterium can't post-translationally modify their proteins that are produced from the gene. So it might be really important to have some sugar molecules or something added to your gene if it's come from a eukaryotic source. But if you place it into a bacteria, which is a prokaryote, you're not going to be able to get that. So you need to use what we call expression vector, such as that it's that's able to perform those tasks. So yeast might come into might be used, or you might actually use a specific eukaryotic cell line which can be transformed. And you can see that the complexity of plasmids can be increased no end. You can keep inserting things into that particular plasmid, obviously up to that particular size limit, but you can put in all sorts of genes like the antibiotic resistance genes. And what do you think would happen if we had a plasmid with an antibiotic resistant gene and we try and grow it on a plate that has antibiotic? Well, it'll survive. It's got the resistance in there. So having that particular antibiotic resistance gene allows it to grow. Okay. So if we think about it, if we have some bacteria and we just transform, so we put in the plasmid even without our insert in there, as soon as the bacterium has taken the plasmid into it, so it's been transformed, the bacteria that actually have taken on that plasmid will have the antibiotic resistance gene. So they're the only ones that will be able to grow in the presence of an antibiotic. So all the bacteria that haven't taken in the vector won't be able to grow, or haven't taken in this plasmid won't be able to grow because they don't have the plasmid that gives them the antibiotic resistance. So we use that to our advantage. So if we have our gene of interest and we placed it into our plasmid, into the multiple coding site, and then we place that, we transform our bacteria with that particular plasmid, and we try and grow it on a plate with antibiotic, whether it be one that has ampicillin, which is a common one, then you'll only get ones that have the bacteria that actually have the vector. Now, does that vector or that plasmid actually have the gene of interest in it? Maybe, maybe not. We know that we won't have the bacteria growing that don't have the plasmid, but whether or not the plasmid actually has the gene that we've tried to ligate into it is another question. And that's where that disruption of that lac operon becomes really important, because it's a selective marker. Okay? And I'll explain that in a little minute. In terms of the old green fluorescent protein, well, all it is, in its simplest form, you can get these plasmids. This one's called PGLO, and it has the green fluorescent protein. So this is that protein from jellyfish. This is the gene that codes for it in here. This is an antibiotic, so beta-lactamase gives an ampicillin resistance. You've got your point of origin, you've got an inducible component here, which is turned on, and we'll explain that in a minute when the plasmid or the bacteria, if it's most of the bacteria is near this particular arabinose sugar, then it's going to allow a lot of expression of this green fluorescent protein. So it's going to be driven the production of that particular gene. So if this was put in, transformed into a bacterium, that bacterium would then be producing the green fluorescent protein, be glowing. Here's just an example of a bacterium, strain of bacterium, DH5 alpha, that's actually had a plasmid that has the ampicillin resistance gene inserted into it, and this one doesn't. So this is what I was saying before. Only the ones that have the plasmid in it will actually grow. There's no growth happening over here. Okay. But the thing to remember here is that you can't allow that antibiotic that's on the plate to wane or become, or not use it. Why? Because that is a selective pressure. As soon as you start saying, I'm not going to put antibiotic on my there are antibacteria there that don't have the plasmid with the ampicillin resistance in them. They have a selective advantage. They probably grow a little bit quicker because they're not having to use extra energy. You put it basically to produce another protein to give it the resistance. So you need to keep the antibiotic in your solution to maintain the correct bacteria. Now, back to the lac operon and how we can use it to actually as our marker. So we know that the antibiotic resistance gene in that plasmid is useful for knowing whether the bacteria actually has the plasmid or not. But how do we know whether the plasmid's actually got the gene that we're of interest in there? Well, you saw in that plasmid diagram the multiple cloning site, which is the bit that we're going to piece in our DNA, sat right in the LAC-Z gene. Okay? So if we have our DNA inserted in there, it's disrupted that gene. It's not going to be produced. Now, what does this operon actually do? Well, the operon's just these groups of genes that polycistronic, so that they're going to be made into one big long mRNA together, and then they'll go through the ribosome in that mRNA, and when the ribosome comes along and detects a start, a stop, it's going to spit out the protein and then keep going along, spit out another protein, etc., spit out another protein as it goes through. It's kind of an efficient mechanism commonly used in bacteria. Now, part of that operon, we have obviously the genes. They're going to be hopefully transcribed and then translated. We have our promoter, and we all know promoters where we're going to have our RNA polymerase come along and do our transcription. But we have this operator site where a repressor sits. And all the while this repressor is sitting on that operator, the RNA polymerase isn't going to come along and be able to do its thing. They're blocked. But if you put in something, and it's usually something that's linked to the function of these genes. So these genes, this particular one here creates an actual enzyme that can break down lactose, beta-galactosidase, so you can use it as an energy source. If you actually have lots of lactose in there, you're going to want that gene active. So you're not going to want the repressor there. So this repressor is made in such a way that if the lactose is present, then it binds to the repressor. By binding to the repressor, it makes a little conformational change and it releases from the operator. As soon as it releases from the operator, then the promoter or the RNA polymerase can come sit on the promoter and shoot along and start doing its thing and start producing the enzyme which can break down the lactose. Okay? So to break down the lactose, this particular gene actually codes for a permease, which will go on actually open a little pore up in the cell membrane of the bacterium and allow a more active transport of lactose into the cell, so it can work even more efficiently in its energy. So that's that whole system there. And this operon actually is sitting in that plasmid. Now, when we grow it in the presence of lactose, we would expect that gene that can break down lactose to be active. And so we have lactose sitting in our clone. But how is that going to allow us to select whether or not our bacteria actually have that? We can't tell whether it's using the lactose or not, whether it's producing the beta-galactosidase. So what we do is we actually have a substrate in there which is very similar to the lactose, so the beta-galactosidase can work on it, but it's modified in a way that when the actual beta-lactosidase actually digests it, it changes the color. Okay, so it changes to a blue color. Right. So therefore, you've got a color selection. So we know if you're getting that color, then you've got that particular that enzyme being active. And if that enzyme's active, well then you haven't put an insert in there because you haven't disrupted the gene, have you? Because remember, we were saying that the multiple cloning site is sitting smack bang in this LAC-Z gene, the one that codes for the enzyme which is going to create the color change. Okay. So if you don't have an insert in there, that enzyme is going to be active and it's going to change the color of that substrate. Okay, so all the colonies, all the bacterial colonies that grow on your plate that change color, we'll know that, okay, there's no insert in there because that gene, that operon is still active. As soon as it's disrupted by your insert, no production of that enzyme, so we know our inserts in there, and we can select those bacteria on the plate and grow them up and multiply them. So grow them up in a broth, and we'll have lots and lots and lots of bacteria that actually have our insert of interest. And then we go and isolate or extract that plasmid, which has our gene of interest, out of the bacteria. And I'll just show that a bit later on. And some of the other points of interest for this operon are nearby the polycistronic gene is an actual regulatory gene. So this is the LAC-I gene. It's not actually part of the operon as such, but it's actually the gene that codes for the repressor. And it's got a promoter of its own, which sits upstream of it, and that's a constitutive promoter. It will always be producing this repressor, because that repressor is needed to regulate the operation of the operon. Because all the while there isn't much lactose available for the cell, the repressor is going to bind to the operator, and it's not going to be wasting its time transcribing the operon to try and digest lactose if it's not there. As soon as lactose comes into the scene, then it binds to the repressor changes, comes off the operator, and the RNA polymerase can go to its thing. Okay. Hopefully that's pretty clear to you, because the whole LAC operon is very, well, it makes it a perfect question for exams, etc. And it is something that you should understand, because not only are you understanding polycistronic genes, so transcription, translation, but you're also seeing how it's regulated and how it has an applied use in terms of recombinant DNA technology. So that's why I'm kind of banging on about it so much. Okay, so here we've got the actual selection happening. So the blue colonies, remember we said if they're blue, that means that that substrate has been allowed to change color, which means that the enzyme was active. And if the enzyme was active, then that gene which codes for the enzyme hadn't been disrupted, so no insert had gone in there. The white colonies, well, that substrate, that F-scar, which is similar to lactose, hadn't actually been changed color. That's because the gene had been disrupted. None of that beta-galactosidase had actually been created. Now that's a color screening. That's the common, most basic blue-white color screening to see whether you've got an insert in there. But it doesn't always have to be based on color. I quite often use plasmids which have what we term a death gene in there. So if the gene's disrupted, then you've disrupted a gene that would normally kill the cell. Okay, so by disrupting it, you're allowing it to grow. So hopefully only the colonies that grow have your gene of interest inserted into them. So there are lots of different ways of having these markers, etc. All right, I can start to see a little bit of yawning. Who wants to take a 5-minute break? Or yeah. Okay. That's recording good. So we spoke about what is that? We spoke about the plasmids, which are one particular type of vector, but there's other vectors. Okay, so bacteriophage. So here we're talking about a virus. These bacteriophages are little viruses which infect bacteria only. Okay, they're actually supposedly the most prevalent organism, far outweighing even bacteria in the world. Okay, they're one of the reasons why the bacteria would have those restriction endonucleases. Okay, because this virus, this bacteriophage, its whole purpose in life is to come along and insert its DNA or its nucleic acid into the bacteria. Okay, so that's why those bacteria have those endonucleases, which we use because they're designed to chop up the viral DNA and make it basically inert. Okay. Now, obviously, just as we could modify the plasmid to transfer genetic material into a bacteria, we can modify DNA in the head here of this phage, and once we've done that, when it actually infects a cell and inserts it in, it's inserting what we want into the actual bacterium. Okay, it's just another way, and I don't think we'll talk too much about it. I mean, there's some benefits to phages. They can hold a bit more genetic material, etc. So plasmids are very simple, easy to use, but there are times and places where phages become useful in their own right. And here we actually can see that the description here, this diagram, is actually quite true to life. When we look at an actual image, electron microscope image of the phage about to inject into the cell. Phages, however, it's a bit harder to manipulate them because with bacteria, we're quite familiar with seeing colonies growing on plates. So one colony hopefully having arisen from one bacterium. But phages, on the other hand, they lyse and destroy the bacterium. So we often do it in a kind of a reverse way. We have a plate that's covered in the bacterium, and we look for the plaques, so where the bacteria have actually been lysed, and that's kind of the negative of actual bacteria working with plasmids. So for all intents and purposes, we consider this to be the colony of the phage. So some of our other vectors, we can have yeast, and yeast can hold quite a lot. We can have bacterial artificial chromosomes and all sorts of expression vectors. I suppose the little plasmid I showed you before was just a cloning plasmid. Plasmids have various purposes, and some of them are made just to copy genes, so to allow that replication of your DNA, and that's what they do very well. But expression vectors are designed so that they'll actually express usually a protein from your gene of interest. So you'll be putting in something that you hope, a gene that will hopefully eventually be translated. So the expression vectors are designed so that they have the essential promoters, etc., to drive the actual, not only the transcription, but the translation of your insert. And that's where we'd probably see most likely the green fluorescent protein inserts. But for you guys, I suppose the general steps of cloning kind of outlaid there. Prepare the vector. If it's a plasmid, it's going to be circular. How are you going to put it into that? Well, usually you have to linearize the plasmid first. So you can either buy it pre-linearized, and that's where a company has come along and it's cut it and created a linear version, and with multiple cloning sites, and all you have to do is come along with your DNA that has the appropriate sticky ends on either end, mix it with ligase with your actual vector, your plasmid in that instance, and will combine with the ligase, and you've got then your intact plasmid with your DNA insert, and then you transform bacteria. And bacteria that need to have the insert placed in them, you can't just do it to any old bacteria. You can, but you get very low efficiencies of the uptake of that particular plasmid. So we make bacteria that are going to take or be transformed, we make them what we call competent, and we fundamentally damage them a little bit, so we make them susceptible, their membrane susceptible to taking in the plasmid, and then that just makes it a more efficient process. Okay. Once it's in there, we can do things to obviously allow the bacteria to replenish or effectively heal themselves and become efficient at multiplying with our insert in there. Okay, analysis of DNA libraries. So one thing we can use plasmids for, I suppose, is creating libraries. And now a DNA library, if you think about it, if we want to know create something that represents all the different genes in a particular cell, we can extract the DNA, which will be one really long linear strand, and then we can use restriction enzymes, so cut it all up. Depending on the enzymes we use, we'll have different sizes. And remember I said with the 4 bases that pretty much whatever those 4 raised to the power of however many sites is going to be, where you have a cut site appear on average in your genome. So you basically chop it all up with restriction enzymes. Then you put this into, so you put it ligate into a load of vectors, okay, and then you put those into various bacteria, okay, and hopefully you'll have a lot of different bacteria, each of them with their own little vector, own little plasmid, and each little plasmid will have its own little part of the genome. So then the problem goes is you've got effectively a library, so a lot of cells, a lot of bacterial cells with all that have a little bit of the genomic DNA in there. You then have to find which bacteria has the book, if you want to put that analogy into use, that you're after. And that's how and then we're talking about screening libraries. So then we can use things like PCR, and we can do PCR on a lot of those colonies, or we can grow the colonies on a plate, and we can use probes. I'll show that in a minute. Same holds true for cDNA libraries, except now with the cDNA library, as you know, cDNA is just mRNA that's been converted into cDNA. So mRNA is fundamentally just the protein, or the genes that have actually been transcribed. Not all the genes of a genome are going to be transcribed, so we've got our transcribed genes forming up mRNA, we convert them to cDNA, we cut them up again, and we put them in to a lot of different vectors which go into bacteria, and once again then we have a library that represents all the different cDNAs which we can then think of as mRNAs. Okay, this is just ways of doing that. It's probably not as important. We covered this with our reverse transcription assay. If we need to screen our library, so find the bacteria that has what the book we're after in, how do we go about that? So here it is, just going through making them again. We've got, in this case, phages, but we could have bacteria. Okay. We've got a lot of them. We have them growing on a plate. Okay, so now we've got plaques instead of colonies. Each one of these will represent one of the phages, which has its own little unique insert, a part of the genome in it, part of the DNA in it. If you place a nitrocellulose filter over the plate, you'll get a little bit of the DNA, a little bit of the plaque, the phage transferred onto it, and then if you come through and you use hybridization techniques, so you have, say, some DNA that matches what you're looking for, but it has a fluorescent signal or a radio label attached to it,